Right now, more than 103,000 people in the U.S. are on a transplant waiting list. Every day, 13 die waiting for a matching donor. Relying on human-to-human donation creates a hard ceiling on how many lives can be saved. The supply will never scale to meet the demand. To bypass this bottleneck, researchers are looking toward xenotransplantation, using organs from another species to sustain human life. Because the human supply is mathematically capped, finding a non-human source is the only path toward an unlimited supply of donor organs. Given our DNA, it seems logical to look at primates first, but science has largely bypassed our closest relatives to focus on the pig. This side-by-side comparison shows why. A pig's heart and kidneys are remarkably similar to our own in terms of physical volume and mechanical pressure. Pigs also reach maturity in months, and the infrastructure to breed them safely and at scale already exists. Their genetics are well-mapped, making them ideal candidates for modification. These factors make pigs the only biologically and logistically viable source for mass-produced donor organs. Connecting a standard pig organ to the human bloodstream triggers hyperacute rejection. The immune system recognizes the tissue as foreign, destroying it within minutes. The attack is triggered by specific sugar molecules, or xenoantigens, that coat the surface of every pig cell. Our antibodies recognize these sugars as an invasion, causing the blood to clot rapidly and the transplanted tissue to die almost instantly. Traditional immunosuppressant drugs are powerless against a response this fast. Success requires a systematic modification of the donor's genetic sequence. Using CRISPR technology, researchers can now edit the DNA of donor pigs before they are born, creating a custom biological profile. Scientists use a knockout strategy to delete specific genes responsible for those immune-triggering sugars, like alpha-gal and Nu5GC. Then they knock in human genes to produce proteins that regulate blood clotting and signal the immune system to stand down. This process creates a biological chimera, an organ that is porcine in structure, but cloaked in human molecular code to avoid detection. Beyond the immune system, researchers had to neutralize a hidden threat, porcine endogenous retroviruses, or PERVs. These are ancient viruses embedded directly into the pig's DNA. While they don't harm the pig, they carry a risk of crossing over to infect human cells. To eliminate this risk, scientists used CRISPR to find and snip out all 62 individual copies of the virus from the pig genome in a single step. Eradicating these sequences removed the final invisible barrier, turning the pig organ into a safe clinical-grade device. Between 2022 and 2024, this technology moved into the operating room through landmark compassionate use cases in living patients. In these trials, the engineered organs functioned immediately. Hearts began pumping and kidneys began filtering waste, without the instant violent rejection seen in earlier decades. These cases provided the evidence that cross-species transplantation is a viable way to sustain human life. Serious hurdles remain. Scientists must still improve the long-term durability of these organs and prevent delayed immune responses that occur weeks or months after surgery. Even with these challenges, the fundamental mechanism has been proven in living humans. Crossing this biological threshold shifts the organ crisis from an unsolvable supply shortage to a challenge of engineering and precision manufacturing.